So, again, we're working on enzymes now. These are going to be biological catalysts. They're going to be responsible for most of the stuff that you think of as you doing things. That is usually an enzyme that's involved. Typically, they're going to be large globular proteins. They're going to be present in every cell, and in fact, even outside of cells oftentimes. So, all those reactions in your metabolism, those are all going to be accelerated by your enzymes. Without them, they would take so long that it would essentially be like they're not happening at all. Okay? Now, one of the things you have to remember about catalysts is catalysts are not consumed or changed by those reactions. So when a enzyme takes part in these reactions, it's going to be regenerated at the end, and it will be the exact same at the end of the reaction. Okay? You'll get your enzyme back. Enzymes cannot force a reaction to occur that would not normally occur. The enzyme is just going to make the reaction move faster in both directions. Okay? So, if we were talking about, for example, you going up the stairs, an enzyme is going to be like something that would increase the rate you can go up the stairs. Okay? It's not going to drive you up the stairs. You would still have to provide energy to go up the stairs. Okay? All right. So tertiary structure of an enzyme is going to play an important role in its function. The structure of an enzyme is directly related to the function. So what you're going to see in all these enzymes is that structure is what the function does. Without the structure, it's no longer going to function. Okay? Which is why like denaturing proteins and enzymes destroys their function. All right. Okay. So again, this is an example of an enzyme working here. Right here, we actually have this hexokinase. And what you're going to see is we have an active site for the enzyme right here. That's actually the part of the enzyme where the catalysis is going to occur. So catalysis is going to occur. It's going to actually act as the catalyst in that site. You can actually see here a zoom up of your active site. And what you're going to notice here is that it's actually going to bind to the substrate. Substrates are just the reactant molecules in an enzyme reaction. Okay? So in a reaction with an enzyme, the reactants are called substrates because the first thing that has to happen in any of these reactions with an enzyme involved is going to be that you have to bind the enzyme. Okay? So you actually have to bind to the enzyme first. This can then allow the enzyme to decrease the activation energy and make these reactions go faster. Okay. Now, some of the cool things about an enzyme here is it has a very specific pattern and size for binding these substrates. This actually means that enzymes have a lot of what's called substrate specificity, that they only bind very specific substrates. So if you were to have a different substrate, different reactant come in here and try to bind to this, if it didn't have hydrogen bonding in the right place, it's not going to bind properly, is it? If it's not the right mix of polar and nonpolar, it's not going to bind properly. If it was too small, it's not going to be able to bind properly and it's going to float away. If it's too big, it just can't fit in at all, can it? Okay? And so this gives them really good specificity for a catalyst. Incredible specificity, in fact. Now, cofactors are going to be inorganic substances, and coenzymes are small organic molecules that will assist in this catalysis. And so a lot of like the vitamins that you take are actually going to be coenzymes that are going to allow for your enzymes in your body to actually function. Without them, your enzymes wouldn't actually function. Okay? So that's a lot of what vitamins actually are. Now, this right here is going to be the different models for enzyme substrate models. This is called the lock and key model. And what you're going to see is the enzyme here is going to be static. It's not going to change from binding your substrate. Okay? So it's like a lock. You slot in the key then, which will be your substrate, and they'll just fit together and you see no changing of either. Okay? This is like a really simple model to understand. It's a really simple model to kind of visualize. It doesn't take a lot of thought processing, but it is also probably the least accurate of the models. Okay, there's a fairly small number of enzymes and substrate pairs where the enzyme doesn't change at all from binding to the substrate. So a bit more of an accurate model is going to be an induced fit model. And what's going to happen here is it's kind of like how I pick things up, right? So when I'm going to pick something up, I form a loose shape of what I'm going to pick up with my hand, right? And I put it around it. And I can't pick anything up yet, can I? So what happens next is I close my hand and mold it around the object, don't I? And that's going to give me a better bind, isn't it? Same thing happens with an enzyme. When you put a substrate in, it's going to loosely fit the shape of your enzyme. And then what's going to happen is it's going to start hydrogen bonding and doing its different intermolecular interactions. And that's going to form around and make so the enzyme fits that substrate better. Okay? And so what you'll see is the enzyme will actually change shape depending on the substrate. So when the enzyme binds the substrate, that's when you get your final active site structure. Okay? Before that, that active site is going to be a slightly different shape. Right? Note that this can also mean that the enzyme shape in general will actually change, binding a different substrate. Okay? And so what you'll see is that when you bind the substrate, your enzyme will actually change shape a little bit, not a huge amount, but a little bit. And that's going to give a better bind to the substrate. And it'll give your actual active form of your enzyme that's actually bound. Okay? Okay. This is actually a model of this. Here we have the previous hexokinase that we were talking about before. And glucose is going to come in. See how it roughly is glucose shaped here? So it's going to start slotting in. It'll start forming the hydrogen bonds. And because these are going to start forming, see the enzyme actually closes around it to form those extra connections. And so that's actually going to change our active site, change our enzyme's geometry. Notice it doesn't just change the active site, but the changes everywhere. But the amount of changes everywhere is fairly minor. It's not a huge change over here, right? A pretty big change over here, but not massive. Very obvious change here. Yeah. Which is your active site. Okay, so how do enzymes actually work to decrease that energy to make things go faster? Okay, think of it a bit like this. Has anybody ever had to walk to something? Like any significant distances? Is that still a thing? Yeah, kind of? Okay. I have to check. I mean, like, you have no idea how often I get no, that's not a thing, Dr. Clausen. We don't walk over hills. So if you had to walk over like a speed bump, right? Is that going to change the how fast it is to get to school? The university? Not much, right? Okay. What if you had to walk over like 2 or 3 speed bumps? Still not a big difference, right? What if I plump down Mount Everest? That's going to change the amount of time it takes to get to school, right? That's going to be like weeks now, especially if you're not good at climbing mountains. Which I'm probably not good at climbing mountains because I've fallen 3 times. Now I think about it, I'm pretty bad at it. So over here we see the original reaction and see how high that activation energy is, right? And so the amount of time it takes to overcome that activation energy is very, very high. It takes a lot of energy to overcome that. Now what an enzyme does is it's going to break this into parts that are going to be smaller than the original energy. And so what we'll see here is this is the first binding part where the enzyme is binding the substrate, and it's just a small amount of energy. And then you're going to get the next part, which is going to be slightly larger, and that's the reaction. Okay? And so there's several ways that it decreases this activation energy from the original activation energy of that reaction to the catalyzed one. But one of the things I want to note is that this got easier to go this way along, right? See that? It's much easier to go along this way now because it's much smaller, right? Much more easily overcome. But if you're going the opposite way, it's also easier to go through, isn't it? Everybody see it okay? So if I take that Mount Everest, knock it down, and just put some speed bumps between you and the school, it's easier to walk to the school. It's also easier to walk back from the school, isn't it? Okay? And so enzymes work in both directions equally. They're going to speed up both directions of reaction. So going to products and products going to reactants. Both directions would be sped up equally by an enzyme. Okay? Okay, so how do enzymes actually work? Well, enzymes are going to bind your substrates. That's going to bring them in close proximity, and it's going to cause them to line up with each other better. Okay? So if you have two substrates that are supposed to be lined up like this to react, an enzyme binds them, orients them, and then they're going to continuously be next to each other, banging each other like that. Okay? Now, in real, when you don't have an enzyme, you don't have catalysts, they can go any direction, right? There's nothing to make them line up. There's nothing to keep them in proximity. They can be bouncing on that side of the room and this side of the room. They could be anywhere, right? And so enzymes, by doing this, that is a large part of how this increases the speed of the reaction. They hold the two substrates in proximity to each other, so they bang into each other more often. And they also orient them each other so they're the correct orientation to have a reaction occur. Because remember that you can't just react from any side, can you? Kind of like me putting my hands together like this. If I tried to do it like this, not going to happen, is it? Right? So you have to remember, you have to have the correct orientation, you have to hit each other, and you have to hit each other with enough energy. And that's the third part that I'm going to start talking about. So if you orient each other in the correct orientation, you keep them very in close proximity, this is going to cut down the energy. The third part is it's actually going to stabilize these transition states. Those are those high energy states you have to overcome. Okay? And so what it's going to do, it's going to do things like, see how this has that magnesium right there? So that magnesium is actually going to take those phosphates, it's going to kind of start separating them on that bond. And it's going to make that bond weaker. Okay? And so it's going to make it so that it's easier to break the bonds and form the new bonds. And it takes less energy to do that. And so it's actually like a little kind of scissors edge that's working its way into that bond so that it can then do the reaction easier, faster. Okay. Now, the other part of whenever we're talking about how fast a reaction occurs is going to be substrate concentration, right? The more concentrated a substrate is, the faster the reaction should occur. And this is, for the most part, true when you're using enzymes as well, okay? But because of the enzymes, we have another factor in there we have to think about, okay? So let's think about it kind of like this, right? We have stairs going up to the second floor, right? And those are a lot faster than trying to leap up to the second floor or climb there, right? Yeah. I'm sure that you appreciate the stairs, right? Compared to trying to leap up or climb up to this. That would be very difficult. So, however, what happens when we have too many people trying to go upstairs? You have to form a line, don't you? And so there's a maximum speed up that we have, a maximum velocity we have for those stairs, isn't there? The same thing happens with the enzymes. So you only have so many enzymes, right? And once you fill them all up, that's going to be your maximum capacity, or maximum activity is what it's called. It's going to be the maximum rate that you can go with those enzymes, where you have them all being used 100%, they're all full, essentially all the time, as often as they can be. Okay? And this is going to be your maximum activity. This is referred to as your steady state. Note that you will still have, this is a velocity, this is a speed, this is a reaction rate. Okay? So this is going to be a steady state where you've hit the maximum speed you can go You're still going to be going more and more product all the time, but you can't go any faster anymore. You have all the enzymes filled. Okay. Now, notice that again, this depends on your concentration of your enzyme. Okay. Below this steady state, more substrate means faster reaction. Okay. It isn't a linear, it's actually a logarithmic, but more substrate means a faster reaction below this. Once you hit the maximum activity, you have all your enzymes full, you can't go any faster. Okay. The example your book likes to use is actually like taking parts and workers and tools for building like a bike, right? And so you have your reactants here, right? Your parts and your workers, right? And so if you have, a million workers, a million parts, you can go pretty quick, right? But a worker working without tools goes really, really, really slow, don't they? Okay? And so you wouldn't be able to go that slow without the tools. And so this is actually what typically is happening in like third world countries actually, is that they have very low access to tools, right? And so they have a really difficult time getting anything done quickly. Okay? And so this enzymes would be like your tools. They'd be the things that speed the work up, right? Okay. And you have a maximum capacity, a maximum activity, and that would be your number of tools that would be dependent on, right? Increasing your workers, increasing your parts amounts isn't going to really change that after you've maxed those out. Okay? Okay, does anybody have any questions there? Okay. This is why, for example, you can get like a country that they're paying like 10 cents an hour for labor, and yet they're underproducing versus a country that is paying $15 to $20 an hour for labor. It's the tools limits, right? The country that's 10 cents doesn't have the tools. Or if they have them, they're broken, not functioning, etc. Very, very common problem, actually, in the world, in case you're wondering. Okay, pH. Enzymes are going to be their most active at a pH optimum. Okay, so remember that your structure is directly equivalent to your function, right? So what happens if I change my structure? I'm going to cause my function not to work as well, right? So remember that we have a lot of groups on here on these enzymes that are side chains that are going to be things that are pH sensitive, right? And we have things like salt bridges. These are also pH sensitive. So when you change the pH, it's going to change its tertiary structure. The pH optimum is going to be when the enzyme maintains the tertiary structure at its most active state. Okay, so it's going to be at its most active. Now, changes in your pH, this can change the nature of amino acid side chains. The carboxylate anions are going to get protonated at lower pHs. The amines, on the other hand, are going to get protonated at lower pHs and deprotonated at higher pHs. And so we would actually see those change charge. These changes can cause the enzyme activity to decrease greatly. So your pH optimum is actually going to be based on the location of your enzyme. Your enzymes end up getting specifically designed for the different parts of your body. So if we're talking about the stomach, that's going to have a very low pH, right? Because it's going to be like HCl, acidic environment. Okay. As we get further away from the stomach and our intestines, we're going to get more and more basic. Okay. So we're going to start as very acidic, pH 2-ish, and then we're going to move to that pH 4, pH 6, and eventually we'll end up in like the liver areas and certain parts of the intestine as an actual basic environment. Okay. Okay. And so this right here is an example of some of these. This is pepsin. It's actually going to work on peptide bonds, breaking up proteins. And it's going to work best in the stomach. Its pH optimum is 2. And so that's a good optimum for the stomach environment, which is about pH 2-ish. Okay. This is sucrase. Its pH optimum is actually 6.2. Notice it's slightly acidic. Okay. And that's because it's in the small intestine, and so you have part of the stomach acid still working its way out in that small intestine environment. Okay, so it's still a little acidic. Urease, this is for urea, as in like urine, stuff that comes out in your urine. This location is the liver, and you'll notice it's a little bit basic, but pretty much physiological pH for most of the liver. There's basic parts of the liver, but mostly it's going to be physiological pHs. This right here is hexokinase. There's going to be all tissues, and you notice it is the physiological pH that we usually talk about, right? The 7.5. There you go. Yeah. And so that's the majority of your environment. Trypsin right here, this is the later parts of the small intestine. This is going to be your other peptide bonds. It's going to help you break apart the rest of those proteins. And this is going to be pH 8 for its optimum. We're getting into the more basic part. This is arginase. It's in the liver, and this is going to be a more basic environment in the liver that typically operates. And it's all the way up at 9.7, which is quite basic, actually, if you think about it. Yeah. All right, so let's talk about temperature. There's also going to be a temperature optimum. For most human enzymes, it's going to be your normal body temperature, which is approximately 37 degrees Celsius. That's why your normal body temperature is that temperature, is because that's your temperature optimum for your enzymes. Okay? Now, above that optimum temperature, your enzyme is going to lose activity due to disruption of the attractive forces. This is going to be what stabilizes your tertiary structure. If you get a high enough temperature, it's actually going to break those completely. And you'll actually get denaturing, which causes permanent damage. Below that denaturing temperature, though, you just have it where it's disrupting the structure, hydrogen bonds are failing, and so you don't have quite the right structure anymore. Okay? And so it'll become less optimal. Again, at high enough temperatures, you actually get denaturing. That's what you get when you cook stuff, right? And if you're ever wondering, yes, you are technically, bless you, little bit cooking yourself in a really hot shower. Okay? Very, very slightly. Okay? Your body's good enough at regulating temperatures that you shouldn't actually harm yourself. But if you get out and it's red for hours, you probably did slightly cook yourself. Okay. Okay. Now, at low temperatures, your enzyme's activity is going to reduce just because it lacks energy. If you get low enough temperatures, you'll start freezing the water around it. When you freeze the water, it can mechanically break the enzymes and your cells. And so that's where you get things like you start turning black and blue because it's so cold, because it's literally breaking your hemoglobin in your veins because of freezing parts. Okay? And then you start getting decomposition very slowly, and yeah, necrosis, and you die horribly. It's really nasty. But if you actually can get that temperature lowered fast enough, you can actually avoid the crystals being able to form and break things because they don't have a chance to move. Okay? And that's where you actually can freeze something and preserve it, if you're ever wondering. Okay? Now, what ends up happening is because you are freezing, you don't have as much energy available to you in the environment, right? And so your reactions stop happening. And so you kind of drift off and you start dying. It takes you a while to actually die, though. Heat typically kills people faster, but it takes very, very extreme temperatures to actually kill them. Okay? Now, anybody ever heard of a... What in the world? Okay. Now, when you're running a fever, what is that actually? What are you doing? What's happening? Why do you run a fever? Mm-hmm. Yeah, you're trying to kill a disease, right? So you have an infection, and your body, what it's doing is, your body has multiple organ systems, right? And it's actually fairly resistant to heat change, changing your temperature, because you don't fail all in one spot. Okay? And so what can happen is your body raises its temperature, And what's hoping is that the bacteria or the virus that's in you can't handle that raised temperature as well as you can, so that you out-survive the virus or bacteria. Now, sometimes that does not happen, and then people die from the fever, right? Sometimes that runs out of control. You are raising your temperature so high that you no longer function well enough to maintain basic bodily functions, like your brain usually is the one that gets cooked. It doesn't actually get cooked. It just fails to maintain the reactions it needs to survive. Yeah. They're too inefficient at that temperature. And so that's why oftentimes like people will cool down the brain area. The heart's another one that you want to cool down. It's a little harder to cool down though, but it can usually operate at higher temperatures. Now, does everybody know that you actually lose the majority of your heat from things like your head and your ears and stuff, and your extremities? Okay. Now, anybody know what kills more people every year? Heat or cold? Cold kills 5 times the number of people as heat in a given year. Yeah. So, yeah. We're actually really good at handling heat overall. As long as you can evaporate water and you can drink enough water, you can handle. You can actually go into an oven and live there. A guy actually did that. One of those magician types, that does the extreme stuff. He had a water pipe going in. He had water continuously delivered in so he could drink. And he went into an oven and stayed there for several days. He actually only needed to come out because he got weak from lack of electrolytes. Yeah. So if they'd known about electrolytes, he could have probably went. But yeah, you can handle really extreme temperatures in terms of heat. You can't handle much in terms of cold. You don't have really great mechanisms to warm yourself up, not as good as you do to cool yourself. So, now, admittedly, humidity can block off a lot of that and make it very difficult for you to actually cool yourself. That's where people get in trouble. They don't drink enough water, too humid out. So you do have to watch out for that, and also overexposure to the sun. So please watch out for those things. Okay, now temperature in terms of if we're talking about things like spoilage, this is actually how we keep food from spoiling, right? We put them in a cool environment, which would be like your fridge or freezer. And so what's going to happen here is the enzymes that are responsible for your food spoiling, i.e. the bacteria need enzymes to do all the work that's going to make them be able to split, breed, and break down the food, right? Those are going to start not being able to operate as fast. And so when you have them in the refrigerator, they're going to operate slower, and that's going to delay spoilage. Okay? When you put them in the freezer, for the most part, most of those enzymes are not really going to function much, right? And so what's going to end up happening is if you put things in the freezer, they can actually theoretically not spoil almost ever, as long as they stay frozen. But they're going to freeze or burn, and their texture is going to change because you froze the ice in them, right? And so realistically speaking, after usually about 3 months, you probably don't want to eat whatever's in there anyways. So you're probably just better off throwing it away, unless the texture has nothing to do with it, and you just don't care about the taste or texture. Okay? But they can last a long time, years, in a freezer, and they won't spoil. Okay? If you go down to a -80 freezer, by the way, those are the things you use for long-term storage of samples. And those can preserve samples for just years and years and years, decades even. You can preserve seeds for, there are seeds that were preserved 4 decades ago that were then used to grow plants of an extinct species. So, again, if you have a bacteria, one of the ways you can actually kill the bacteria and make it so that you can decontaminate something is by boiling the water right. And so that's how they do things like decontaminating tools for surgeries, decontaminating tools for dental, and they do things like decontaminating your water. You can boil the water. Boiling the water will kill all the bacteria in it. Now, one thing I would like to point out is that if you get like something in your fridge and it's full of mold or bacteria, and then you cook it, yeah, you'll kill the bacteria and mold, but that doesn't mean you've gotten rid of the poison that was in it. Okay? And so people make this mistake all the time. They'll take like rotted food and then they'll cook it and go, I've killed all the bacteria. I'm going to be okay. They eat it and then they get really sick, right? And yeah, you did kill all the bacteria, but again, there's things in the bacteria that are poisonous. If you don't otherwise wash those out in some way, you're still going to get sick. Okay? And you can still die from it. One of the leading causes of death all throughout human history, by the way. Yeah. Okay. Let's talk about inhibitors. So enzyme inhibitors are going to prevent active sites from interacting with the substrate to form the enzyme-substrate complex. So some inhibitors can cause enzymes to lose catalytic activity temporarily. Others can cause enzymes to lose activity permanently. So reversible inhibition is where it's going to lose activity temporarily. It's going to be reversible. It can go backwards. And so the inhibitor is going to cause the enzyme to lose catalytic activity. That's what an inhibitor is. If you remove the inhibitor or lose concentration for the inhibitor in some way, then your enzyme is going to become functional once more. Reversible inhibitors can be several different things. Two of the major ones that we're going to learn about today are going to be competitive or non-competitive. So a competitive inhibitor is going to bind into the active site. It's going to bind the same place the substrate would. And so it directly competes with the substrate. If an inhibitor binds there, the substrate can't. If the substrate binds there, the inhibitor cannot. Okay? And so they just compete with each other directly. If you increase concentration in your substrate, you can actually overpower the inhibitor. Okay? All right. Everybody good with that one so far? Can you guys see what the figure says? This is the inhibitor binding, right? If the inhibitor comes off, we can have the substrate bind. Okay? And vice versa. Okay. If the substrate binds, it can produce product. Okay, so non-competitive inhibitors, these are not going to compete with the substrate for the binding spot. They're going to instead bind onto an external site right here, and that is going to cause it so your substrate can no longer bind properly. So this is going to be a non-competitive inhibitor. So when the inhibitor binds, you're going to get so the substrate cannot bind. Okay? When the substrate here binds, it can go to product unless the inhibitor binds, in which case it may get knocked off or it may not be able to bind the inhibitor. We haven't gone into that stuff. But what's going to happen here is you're going to bind with the inhibitor. This is going to change your active site. And so the distorted active site is no longer going to bind to the substrate. Okay, so in an irreversible inhibition, the inhibitor is going to form a covalent bond to an amino acid side chain on that enzyme. And so with the inhibitor covalently bonded in the active site, the substrate is going to be excluded from the reaction, catalytic reaction, and will no longer be able to use that enzyme. So it's going to permanently bind when you have an irreversible inhibitor, that enzyme, and no longer allow the substrate to bind, no longer allow that reaction to occur. Now, regardless of your method, irreversible inhibitors will permanently inactivate enzymes. This is actually an example of an irreversible inhibitor, and you may have heard of it. It's called penicillin. Hopefully everybody's heard of that one. Thank goodness. Okay, and so this is an irreversible inhibitor. Penicillin actually binds to the active site of an enzyme that bacteria use in the synthesis of their cell walls. And so if you no longer have active enzymes that can build cell walls, you can no longer multiply and grow, right? And so it blocks the bacteria from being able to produce more bacteria and, in fact, even repair their cell walls efficiently. Now, when the bacterial enzyme bonds to penicillin, the enzyme is going to lose its catalytic activity, and that growth of your bacterial cell walls is greatly going to slow. Okay? Now, again, without proper protection, those bacteria can't survive. Your infection ends up stopping because your body gets a chance to kill off all the remaining bacteria. Okay? Now, one of the things I would like to point out here is, first off, there's a bunch of penicillin derivatives, in case you're wondering. Penicillin is probably considered the most important and antibiotic that has been discovered. There's a few ones that have been invented. These are called broad-range antibiotics. They work on all the different bacteria. There are one bacteria, though, that have become immune to penicillin. They've developed ways of countering penicillin. Okay? And so these are called antibiotic-resistant bacteria. Right? One of the things I want to ask you is, this works against bacteria. It's an antibiotic, right? If you catch a virus, like a flu, cold, COVID, and you take penicillin, does it do anything for you? Absolutely nothing. Does absolutely nothing for you. Viruses don't have cell walls. They don't have the enzyme to target. It is not magic. It does not help you overcome a cold, flu, COVID, nothing. Okay? It has to be a bacterial infection. Okay? Now, if you take penicillin and you aren't killing off all the bacteria, the bacteria is going to get a chance to adapt to it, right? And so when people take penicillin and they don't need it because they have a cold or flu, that's actually really problematic. Okay? So if you see anybody that's taking penicillin, we're trying to protect penicillin right now because we don't actually have any really, really broad scope antibiotics that can actually fully replace penicillin yet. Okay, we actually have a few candidates, and maybe they'll work, but they too will end up having resistance developed against them. Okay, so doctors try to conserve these and protect these, but what ends up happening is people freak out. I'm really sick because of cold, flu, etc., COVID, virus. They take antibiotics, they do nothing against the viruses, and then they end up breeding and creating antibiotic-resistant bacteria. Which then means that once that starts spreading, we don't have anything to counter. Okay? And so please be very careful about that. Don't just take an antibiotic and just take it for no reason. Okay? Now, if you're going to take your antibiotic, this is the other end of that. Don't stop before the doctor's prescription says to stop. Okay? So what you're doing when you take an antibiotic is you're flooding your system with this inhibitor, right? And it's going to destroy all the enzymes for those. If you stop flooding your system with the inhibitor, what's going to happen? Well, they're going to start making more enzyme, right? And that enzyme is now going to be active and they'll spread. So if you keep doing that, so let's say you had a 5-day prescription, you take 2 days, you feel better, don't need it anymore. And then you get sick again, don't you? Because the bacteria was never actually killed off. Okay? And then you take another 2 days. Oh, I feel better again. And you'll keep going through these cycles, right? And what will happen is eventually that bacteria is going to evolve and it's going to become antibiotic resistant, isn't it? You're training it. Okay? So if you're going to take antibiotics, you got to go the full course. Okay? You got to decimate, destroy that bacteria. Don't allow it to recover. Okay? And if you don't need it, don't take it. Now, do you know why they don't just flood everything all the time with antibiotics? Your kidneys get damaged. Yeah. So because you're putting so much of this substance in your bloodstream, your kidneys are working really hard to remove it. And so over time, your kidneys will get damaged. Which is why they don't just go, just take it for 30 days. We'll just put you on this for half a year. No more sickness. It would kill you. Very likely. It wouldn't kill you right away, but it would damage your kidneys. And if you're older, it would probably kill you. Anybody have any questions there? Okay. Okay. Interesting little side note. There are different types of antibiotics. Most antibiotics are not what are called full spectrum. So there are many antibiotics that are only good against certain types of bacteria. Okay. And because it's targeting specific enzymes. So you got to be very careful. If you're taking an antibiotic and you're not getting better, talk to your doctor. Like, this is not an actual like rare thing that happens. This is quite common actually, where a doctor will assign you an antibiotic. They didn't bother doing the check to see what exact bacteria you have, and it doesn't work. And it's because you have the wrong type of bacteria. Okay, that's happened like 4 times to my mother, and I'm like, talk to your doctor and tell him to test the damn bacteria. Stop being lazy. Just test it. Yeah. And it's because they try to conserve these full spectrum antibiotics only if they need them. Because if they use them all the time, they won't be useful in the future. Yeah. And we have to sort of delay the inevitable uselessness of them as much as we can. Okay. Anybody have any questions? All right. Study guide again. Everybody know where to find PowerPoints, all that? Fantastic. If you don't know at this point, please talk to me. Okay. We're on chapter 11. My goodness. All right. So, on Wednesday, I will not be here. Everybody remembers that, right? Okay. Okay. Thursday, I will not be here in the morning. Okay. And so I had released an email, and what you all will do is you'll end up watching the video for the rest of this chapter. Okay? And that will be your lecture for that day. Yeah. I have a follow-up to my eye surgery on Thursday. Okay? It's my post-op. Make sure nothing weird happened. I still have those eyes. Okay. I think I'm going to move that online, actually. So I'll be here Thursday afternoon, but I probably shouldn't have all the smoke in the lab and stuff getting into my eyes. Yeah. Yeah. So is everybody okay with that if I end up moving it online? Yeah, I'm going to move that online. Okay. You can write out the scientific method and submit that. Okay. So this one, we're going to be talking about nucleic acids. Again, these are going to be things like DNA, RNA, right? So very, very, very large molecules. Now, please don't make me talk over people so much. Thank you. Now, DNA, this is going to be found in your nucleus of your cells. It's going to contain genetic information, which is going to be called your genome. The gene is going to contain information to make a particular protein for the cell. The human genome contains about 20,000 genes. Different cell types will express different parts of their genome. So not every part of your genome is expressed in all parts of your body. Okay? In fact, some of them may not even express at all, really. You can have genes that are dormant that don't really express. Nucleic acids like DNA and their counterparts, which are ribonucleic acids, which is RNA. These are very, very large molecules that are made of repeating blocks. These are called nucleotides. And so, yes, they are types of polymers. The monomer would be the nucleotide in this case. Okay. If you explore the structure of a nucleotide, it's going to be the starting point for your nucleic acids, for your genomics, and study of genes and their interactions. Okay. Now again, remember, proteins are going to be strings of amino acids. Nucleic acids are going to be strings of molecules, which are called nucleotides. And so nucleic acids are going to be created from a set of 4 nucleotides in a given sequence. You probably all have heard this, like ATGC, right? So nucleotides are going to have 3 basic components. we have what's called a nitrogenous base. That's this part here. That's a version of that part at least. We have the 5-carbon sugar, and then we have a phosphate functional group. That's that same phosphate that you remember from like phosphoric acid, phosphate, the anion, all that stuff. Okay. You'll also recognize that 5-carbon sugar in a bit. All right, so this all together is a nucleotide. Okay, you have your base, sugar, and phosphate. Okay. That's the base unit of DNA and RNA. Okay, so these are 2 of the nitrogenous bases. This is 2 of the versions of the nitrogenous bases. Oh. Hello. What are you doing there? Okay. Now, this is the purine. Purines have a double ring. This right here is the pyrimidine. It has a single ring. Everybody have that okay? See how they're aromatic rings? They contain 2 nitrogens per ring. And again, purine, double ring, pyrimidine, single ring. Okay. Always kind of remember this as pyramid, pyrimidine. Pyramid comes to a point, single point. Now, this right here is going to be the purines. So adenine, which is A, that was the ATGC, the A part. This is going to be, do I have anything I can hit this with? No. Yeah. Okay. Be careful if you're allergic. So, your purines right here, this right here is adenine. It's going to be the A from the DNA and RNAs. This right here is guanine, your G. This is going to also be in DNA and RNA. Okay, notice the differences here. Here you have an amine versus a carbonyl. You all okay? Maybe I have a book they can swat it with? Okay. Okay. Okay, so those are your purines. Let's talk about pyrimidines. This is going to be your CT in DNA. So this is cytosine, that's your C. This one's thymine. This is your T in DNA. Now note that RNA does not have thymine. Instead you have uracil. Got it? Cool, good job. Woo. Got it. Okay. And so again, RNA only has U, right? DNA has T instead. Okay. Does anybody know how they match in DNA? So what matches with A? T, you got it. What matches with G? C, yeah, you got it. Okay. Does anybody notice something about that? C matches with G, right? What do you notice? A matches with T. So every matching set has 3 rings, doesn't it? Okay. 1 ring always matches with a 2 ring. Okay. And so that always gives you a 3-ring set. Right. Okay. So this right here is your sugar. This is ribose right here. You may recognize that from the sugars, ribose. Here's the first and second position. Now, DNA actually stands for deoxyribonucleic acid, right? And so this is going to lack an oxygen on this second bond here. See how we've lost an oxygen there? And so that's deoxyribose. Removed an oxygen. So the R in RNA is for ribose. The D in DNA is for deoxyribose. Right there. Otherwise, they are identical. They don't have any difference between the nucleotide for DNA or RNA other than the deoxy. Okay? Okay, so these are joined by a condensation bond. You'll notice that it's going to be very, very similar to our same condensation which made our glycosidic bond before, right? And so here we have the adenine base. It's going to undergo a condensation reaction. This is going to form our glycosidic bond, same as before, right? This right here is the beta. Okay. And this right here, when these two are joined, this is going to be called a nucleoside. So without the phosphate, and the nucleotide is called a nucleoside. The easiest way to remember that is this is the side part to a nucleotide. The phosphate is part of the backbone. Okay? And so you typically call the phosphate portion a phosphate backbone in DNA. All right. So now we're also going to do another condensation reaction. This is going to join the phosphate. It's going to be joined to the 5th carbon portion here on that ribose. That's going to give us our adenosine monophosphate, which we call AMP. M for the mono, right? P for the phosphate. Okay. Adenosine for the A portion right here, the base. Okay. Now, anybody ever heard of ATP? Guess what that has? Triphosphate. ADP has diphosphate. Yeah. And so ATP, i.e. the power source for your cells, right? That's triphosphate. That's just 3 phosphates over here. ADP would be 2 phosphates. MP, 1 phosphate. Okay. You have the same sort of thing when you're talking about ATP. You actually have those for GTP, CTP, UTP, all those. Okay? You can get the same things. So, right here we have AMP. That has the OH there. If we have the DNA version, it's going to be the deoxy. And so that will be a lowercase d, and that would be DAMP. Okay? If you had the guanine here instead of the adenosine, this right here is going to be GMP, right? If you have the DNA version, it's going to be lowercase d in front of it for deoxy. Okay? Now, the only place that this is really different is going to be where you have UMP. You don't have a DUMP because U doesn't happen in DNA. It only happens in RNA. Instead, what you have is a DTMP because you have the T in DNA, don't you? Okay. Again, nucleotide is going to contain all 3 of these. Nucleoside is only going to have the sugar in the base. Okay. Remember that the M is for the number of phosphates. And so again, you can have a GTP, guanine triphosphate. And that again can be used as power in your cells just like an ATP can. Okay? Okay. These again are the full names versus your nucleoside versus your base right here. Okay? Anybody have any questions there? Okay, fantastic. Lose anybody yet? Okay, so many nucleotides linked together. This is going to form a nucleic acid. Nucleotides are going to be linked through what's called phosphodiester bonds. And what I want to point out is, again, this is going to be another condensation reaction that happens. And what you're going to have is this phosphate here is going to be linked to the 3rd position here on that sugar. Okay? And again, that's going to be right there. So what we see here is, do you notice this looks a lot like an ester right here, right? We have a carbon bond to an oxygen, bonded to something else, right? Yeah. So it looks a lot like an ester. It's just an ester with a phospho, right? Phosphate there. So we have a phosphoester, right? And there's 2 of them on each side of the phosphate, isn't there? And so a phosphodiester. Di for 2, right? Yeah. Makes a lot of sense, doesn't it? Okay. This is actually the start of like a DNA backbone right here. Okay? Yeah, be DNA. Because it's deoxy. Okay. Now the oxygens in the phosphate, notice that these oxygens are connected between the 3' end right here and the 5' end. Everybody see that okay? Okay, so that gives us a natural directionality to these backbones, right? And so we actually have a convention for reading these. We typically read these from the free 5' end to the free 3' end. Okay? And so again, from the free 5' end right here to the free 3' end right there. That's actually how you would typically read along these. Okay. And so this would represent phosphate sugar with the side chain right there, which is your base. This then is going to be condensed down, and this is just letters. And again, it's representing the same thing each time. And again, you read it 5' on the left, 3' on the right. Because in English we read left, right. Okay, so the base sequence in nucleic acids is stored as DNA in your cell's nucleus. It's going to hold the code for the cellular protein production and RNA production, of course. A few key discoveries began in the late 1940s-ish, and this actually led to the structure of DNA. Erwin Chargaff, noted that the amount of adenine is always equal to the amount of thymine. So whenever he took DNA, took it apart, A and T were always in equal amounts. And then he also noticed that C and G were always in equal amounts whenever he chopped it up and took it apart. And so the number of purines always equaled the number of pyrimidines as well. Now, remember, at that time, they didn't know anything I had just told you, right? So if you didn't know anything about this, you wouldn't know that a purine always matches a pyrimidine, do you? Okay? This is where that came from. All right. So what's the secondary structure look like? Now, your book still has this in it, which is a little... So, James Watson and Francis Crick were given the Nobel Prize for discovering the secondary structure of DNA. They did not actually. They actually stole the results from two women that worked next to them. They're the ones that actually made that discovery. They just didn't get published before Watson and Crick did. Now, so they have a bit of a checkered history. Watson actually just died like a week ago, in case you're wondering. Now, having said that, Watson and Crick did do some really good work in that field, and arguably probably were Nobel worthy in and of themselves. If you're talking about who predicted the structure, that's actually Linus Pauling that predicted it. If you're talking about who actually discovered experimental proof, that's the two ladies that worked next to their lab. Yeah. They're quite famous in and of themselves. I'll let you look that up because very worthwhile documentary if you ever get a chance, okay? Anyways, they discovered that the double helix was the structure. It's two helices that wrap around each other, right? And we'll see a picture in just a little bit. In these helices, they're actually going to run in opposite directions. So those backbones are going to run from the 5' to 3' in one direction, and then the 3' to 5' in the other direction, okay? And so they'll run in opposite directions. Each rung on this double helices is going to be one base from each of the strands that overlaps. And it's going to be a 2-ring structure for the base, overlapping with a 1-ring structure. Okay? Now, the two bases are going to overlap in these ring structures. They're going to overlap so that they form proper hydrogen bonding between the two. And they're always going to end up being the same length on every single rung of this base because they're always 3 rings long, right? Okay. And so AT and GC, these are going to be called the complementary base pairs. And the reason that they're complementary is A and T have 2 hydrogen bonds that they form. And so they'll always form a set of 2 hydrogen bonds that are correctly aligned. Okay. If you look at then G and C, they're going to form a set of 3 hydrogen bonds that will correctly align each time. Okay. The DNA in one human cell is going to contain about 3 billion base pairs. This right here is the 2 hydrogen bonds being formed. See how they form those 2 hydrogen bonds and they're overlapping in the correct space. If you ever got, say, an A trying to overlap with a C, what you would notice is these would not properly align. And so you would not get good hydrogen bonding happening. Okay? What would happen if you tried to overlap like an A here with a G? That wouldn't be this, would it? Wouldn't be 3 rings, that would be 4 rings. And so it would bulge out in the structure, it wouldn't properly overlap, would it? If you try to overlap a C and a T, they'd only be 1 rings. They wouldn't quite get to each other, would they? And so they wouldn't bind with each other. This is what DNA looks like when it's given a sort of cartoonish structure. So you can see inside of it here. What you'll see is, again, the double helix structure right here. Everybody sees that okay? Here we have AT overlapping, and they're giving us our 2 hydrogen bonds to keep them together. Okay. Here we have a C and G overlapping. They give us our 3 hydrogen bonds to keep them together. Kind of an interesting thing that happens here is you have these fairly nonpolar rings that overlap like this on top of each other. That actually does contribute to holding this DNA structure together, in case you're wondering. So it's not just the hydrogen bonds here that contributes. It's also the fact that they're overlapping on top that contributes. Okay? And so that's actually a lot of the reason why you need that 3-ring structure to properly overlap on top of each other. Okay? They slowly turn as you go down like this. Again, here's your phosphate backbone right here. It's a sugar phosphate backbone. Okay. And you'll notice here that it has 2 different groove size. Can you guys see that this is like a big groove? This is the small groove. Major, minor grooves. Okay. It's really important for function. Now, you may have noticed that these DNAs, these were pretty dang big, weren't they? Okay. And so what you would see is if you actually had these 3 billion base pairs in one human cell, if you stretch these out into a straight line, like you would think that they'd be in your cells, these would be 6 feet long. Okay? That is not going to store in your cell properly, is it? Not at all. Okay, so what ends up happening is, you all have experienced this. You ever taken your cords and you put them in your bag, right? And then they all get tangled up everywhere? And you try to fold a cord and it starts twisting back on itself and doing weird stuff. Or like a hose, you may have done this and really had to fight it. You start trying to wrap it up and it starts trying to go back on itself, right? And then you have to twist it with your hands. Okay, that's actually called supercoiling. That's what that's doing. And so DNA does the same thing because it has this double helix structure. It experiences strain when you start folding it in. You start coiling it around stuff. So this DNA, that helical twist, is going to cause further twisting, called supercoiling. This is going to coil up around, and it's going to form an even more compact structure. It uses histones, which are these little structures right here that look like little balls. They're kind of like your, those little plastic things that you can wrap cords around to keep them organized in your bag. They're like those for DNA. They keep them well organized. And so they'll wrap around histones to form larger structures, the supercoiled DNA. Eventually, they'll form even larger structures with the supercoiling. That'll give you chromosomes. That is going to give you 46 pieces that are supercoiled around the histones. And again, that would be your chromosomes. You may have heard of 23andMe. Each parent contributes 23 chromosomes to give you a total of 46. So that's your 46 chromosomes right there. Does anybody have any questions? You all have a wonderful day. I will see you after the break. Okay? Nope, not next Tuesday. The Tuesday after, yeah. Yeah, we had a long, long break. Okay?